This commercially available test allows for the quantitative analysis of parasitic eggs, oocysts, and larvae found in animal feces. Here we perform the Flotac basic technique on a sheep fecal sample. Weigh 10 grams of feces and place it in one of the two plastic containers. Using a graduated cylinder, measure 90 ml of tap water and add it to the sample, dilution ratio 1:10. Homogenize the contents with an immersion blender and filter the solution through a fine mesh strainer to remove larger debris, and transfer the filtered contents into the second plastic container. While keeping the suspension truly mixed, collect 6 ml of the filtrate with a serological pipette and transfer it into a test tube. Centrifuge the solution for 3 minutes at 1500 rpm. Discard the supernatant by carefully pouring it off into a sink while keeping the pellet in the bottom of the tube. Refill the tube with flotation solution, restoring the initial volume of 6 ml and resuspending the pellet into the solution. Assemble the Flotac device, see the figure, and fill the chambers of the device with 5 ml of the resuspended Faecal solution. Rotate the Flotac to the closed position and centrifuge it for 5 minutes on 1000 rpm. After centrifugation, loosen the locking key and rotate the device to the reading position. Place the Flotac on the microscope, affixing it with the supplied adapter. Examine each reading chamber at 10x magnification objective and count all the parasitic form using the handheld teleclicker to record the number of organisms seen. The total number of eggs counted is then multiplied by a multiplication factors that varies depending on the dilution ratio used. Using the dilution ratio of 1:10, as previously mentioned, would require a multiplication factor of 2, total number of eggs counted per 2, to obtain the number of eggs per gram of feces, EPG. This same process may be used for oocysts and larvae to obtain the OPG and LPG, respectively. Each Flotac device has two separate reading chambers, allowing for samples to be processed one of three ways. The basic technique involves processing the sample in duplicate with the same flotation solution. The dual technique uses two different flotation solutions for the sample. Finally, the double technique allows two different samples to be processed at one time with the same flotation solution. The dilution ratio should be adapted to the expected number of parasitic elements, PE. The count results with a dilution ratio 1:10 are accurate when the number of PE is under 500 per grams of feces. When the PE per grams are greater than 500, that frequently occurs in sheep and goats, it is advisable to dilute the sample more.